Hello, everyone, and welcome to the second lecture in the series of AI in healthcare and medicine. My name is Dr. Abdullah Shaheen. I'm a clinical associate professor of medicine at Loyola University, and today I'll be giving you a brief introduction to the in silico research world and see the amalgamation of translational, biomedical, and big data research coming together and changing how we actually perceive research going forward. So for today, I'll be talking on a few domains that I personally work on and how they amalgamate to become the in silico research model that I work on, and also introduce what basically bench research on a chip or in silico research means, and also talk about the roles of machine learning in future immunology and how is that going to be shaped by our current technology. I have nothing to report in terms of conflicts of interest. In terms of the platforms I'm going to be presenting today, most of them, if not all, are open source platforms, amazing tools, and I encourage you all to kind of play with them in a sandbox mode. So I personally have worked on sepsis and immune modulation as my basic science and translational science interest, and I'll talk about how sepsis came to be what it is today. From the word decay in Latin, it progressed over years and years of what they tried to describe initially on the gross clinical appearance of people just deteriorating in front of their physicians or the community's eyes, and what is driving that decay, the physical decay that they are observing on their patients. As we came to the 19th and the 20th century, and we started understanding the body as a machine, different systems, and we started talking about the system's response to this process. So we've shifted our focus from just the general description to the systems-based understanding, as we actually started also introducing the germ theory and started understanding the main culprit of what drives sepsis is. We started talking about the interaction between the host and the bug and the process itself. And today we have the SIRS and SOFA tools that we use to classify the severity of these processes that we are observing and quantify them in some way. And today we talk about immune phenotypes and endotypes after our advanced experience with things like COVID and other clinical diseases that we noticed significant immune profile differences in different hosts. Also in immunology, we started having a process of resistant to reinfection understanding, where we saw people recovering from an infection and never getting it again in its next wave or next round of infection. We started talking about modern immunization introduced by Jenner for cowpox as a way to protect from other pox diseases, to what actually works on the biological level, the cellular and humoral immunity, and how we understood all these processes, different processes for each of them, and how they work to produce the effect that we are trying to obtain, to developing immunoglobulins and understanding the MHCs, TLRs, and PRRs, and how they actually play a role in the process and how we can target them specifically to alter the immunological profile or the response that we are trying to either augment or prevent from happening. And eventually, we are now in the age of genomics and transcriptomics that actually also shape how we, from the beginning of the immune system building, can actually either alter or understand the risk of developing something with a specific disease or a specific infection before even it happens, just from understanding the genomic profile of that person. The history of data science is a bit more interesting. We've had statistics for the entirety of our history, at least the written history. Even in our cave times, we used to actually have scratch markets for quantifying some of the things that we did in terms of harvesting, hunting, and others. So we always kept a numerical representation of what we are trying to do. There's always this interest in quantification. Statistics was used heavily in the Sumerian and Assyrian era to actually for plans of taxation mainly, and then eventually also for harvest planning and modeling for what next year's needs will be like. It wasn't until the 1700s that we started having methods at our statistical to predict the needs of the future or the modeling of the future. And actually we started in what today is the UK, where they actually were trying to manage the pension funds and understanding how much they need to collect from folks to plan for their retirement, knowing that they will live for a certain time and a certain profile of people, and understanding how they can calculate and predict that became of a big interest of them, and that's how most of the statistical methods started to be born. We continued to refine our methods. It was all manual calculations up until we started having computational tools, talking about the beginning of the computer age, the machine computer age. And that was in the 1940s, 1950s, when we started actually building mathematical models that can do that in a quicker way, in a bigger sets of data. And then by the 2000s, we have accumulated tons of data that is historic, that is really highly grained or highly refined, and started developing these models that instead of running on thousands of rows, can run actually 40,000 columns with millions of rows and so many different data points, interactions. And that actually brought us to this age of AI, machine learning. It was the accumulation of the data we had throughout the history, the cleaning of this data, the interpretation of this data, and then using these statistical models that we actually have been also refining on the other end. And the thing that is timeless is the domain expertise in any field that you are trying to apply data analytics on. This is the core of any of the work that you try to do today and you should be trying to do in the future. It's the domain expertise that is timeless, and it's your powerful tool to be relevant in the future. So not only banking and social media and shopping were the things disrupted by data science. Biomedical research was among the early to be actually dampened with, and data science research, starting with some predictive modeling using machine learning, as soon as we developed these tools, because we have also accumulated tons of data over the years from our research, from our clinical practice, that we can use to do these modeling and predict outcomes that we are trying to look for early on, regardless of how successful we are with these tools or not. Single-cell omics in bioinformatics is another field that we have excelled very well over the last 30 years. Our understanding for the single-cell functions, the pathways, have really improved with all these kind of boring research that we have done. So thank for all these PhDs who dedicated careers to a single pathway, a single molecule, because these folks actually give us the most important part to understanding the single cell functions, and it actually drove the form of the field of bioinformatics forward years at a time, because as the time the computers came to work on this data, our scientists have actually done tremendous amount of work on it. And of course, tribute to all these mouse models and animal models that have to be sacrificed for this. We actually have developed a lot of interest in we have actually developed a lot of data points that actually will help us use computers to simulate and hopefully save more mouse and animal models from being sacrificed on the pathway to learning more about our human body and immunology and biology. Biomarker discovery and validation is something that data science actually worked on significantly in cancers and other fields, and we'll put a few examples as we go forward. Clinical decision support systems, CDSSs, are things that are also developed because of data science advancement, as we spoke in the first lecture. Nowadays, we still see rudimentary forms of data decision support applications, but it's a matter of time, and it's going to be very soon that we see much more advanced CDSs working with us on our clinical side. Some of them already are manifesting in your clinical practice as you Google more stuff. Now ChatGPT more stuff or Claude more stuff, whatever LLM model you use, you are doing some sort of a support for that way in a manual way that is also still away from the clinical data you are trying to apply it on. But it's not going to be far from today where platforms like Epic and others will be introducing these AI tools to help you know what to order, how to look for differential and such. And deep learning and AI are the key words nowadays, the hubs of interest, the hypes of media, and these are things that certainly have a huge role in medicine going forward. And we're going to have talks for clinical applications today are focusing more on the biomedical research and in silico research applications of AI and deep learning. And uses in the contemporary research are abundant already. So cancer immunotherapy has been really accelerated by machine learning, really analyzing all these immune signatures, predicting patient responses, checkpoint inhibitors. CAR-T therapies have been really shaped by AI and machine learning algorithm research, guiding us to the dream of precision medicine closer. And all these CAR-T therapies that we have today have something to do with biomedical research using AI and machine learning. COVID-19 pandemic was a great turning ground for us to use the data we have to rapid understanding of immune responses, biomarker predictive of severity, vaccine efficacy, and using AI and machine learning. Pfizer researchers were actually were able to put down the data needed for the application in less than 24 hours, and stuff that would have taken them weeks to months to actually prepare for the submission of FDA and other tools. There's an urban legend of them using AI to predict the mRNA molecule shape that has not been validated, but again, it's something that you can actually start using today as a starting tool for your whatever research you want to do, to use AI and biomedical modeling to predict how certain molecules would interact in silico before you actually go and test them on animal or human subjects. Sepsis endotyping is another byproduct of our research in COVID times, where we started using AI-driven analytic tools looking at immune and clinical data to create patient subgroups with distinct immune responses, which will lead us to a different pathway of how we treat these immune endotypes. So from the ARDS developing in lungs from COVID to other application, we are seeing AI-driven tools helping us really distinctively say who should get what early on in the treatment phase. And even in drug design, nowadays we have AI-driven tools that generates the medication, both identifying the biological target and the therapeutic compound, such as rentosirib or rentorcetab, which is the first investigational drug that is 100% designed using generative AI. And this is something that's being pushed to through the pipelines now for approval, and of course expedited by a lot of other AI application tools that we have in our hands today. So after I mentioned a few examples, let's hone in on what in silico research means, how we can navigate the future of biomedical research going forward. So in mathematical and computational modeling, we have different systems. Systems biology models are basically what describes dynamics involving, for example, in sepsis, inflammatory cascades, cytokine storms, pathogen-host interactions, immune dysregulation. I'm using sepsis as an example, but of course it's universal to any physiological process or a pathophysiological process in the body. And these come from our understanding of what biology is. And always what we try to do is alter this biology using different technologies: gene alteration, gene deletion, gene addition, pathway blockade, molecular alteration, receptor blocking or augmentation. All these actually create to us new data that we use computationally to produce new hypotheses, push back to biology, get new insights, and so on. So we keep altering and testing and creating more data. So it's a virtuous cycle of better understanding through computational integration into biology. So these are three common modeling ways, and I'll talk briefly about each one of them. So we have agent-based modeling, we have ordinary differential equations, and we have physiologically based pharmacokinetics modeling. So agent-based modeling, my personal favorite, and it's maybe easier for me to understand, it's basically computational modeling to simulate actions and interactions of autonomous agents to assess their effects on a system as a whole. Autonomous agents are special computer programs designed to work independently. These could be anything from a cell to an individual to a certain variable. Basically, it just tries to simulate an entire world based around a certain agent. It actually captures heterogeneity among the agents. It explores emerging phenomena. It helps experimentation with policy, strategy scenarios. So it's very helpful in public health approaches and community level approaches, and can really handle complex and adaptive systems. It really needs a lot of computational complexity and power as a weakness. It depends on extensive data modeling. You need a lot of data to do that. And it's really hard to interpret the outcomes sometimes. So keep that in mind where you actually are trying to create an entire world and simulate an agent-agent interaction and see how could be altered to alter the outcome or how they interact with each other. A few good ABM tools. You have NetLogo. It's user-friendly, very popular in academia. AnyLogic is a commercial software. Of course, MesaKit for Python is an open source, good for large-scale simulation, very helpful to use. Repass is a flexible, powerful Java-based firmware to also do these simulations. So this is an agent-based modeling in a nutshell. The second is ordinary differential equations, ODEs. In bioinformatics, these are often used for describing relationships between unknown functions and its derivatives. I'm sure these words bring PTSD to a lot of us who focused on biology and forgot a lot about mathematics. But it's quite useful, especially for us in medicine, in gene regulatory network exploration, so how mRNA and proteins change over time. So it has a lot of applications in bioinformatics, from gene expression data fitting. It can really help you to kind of fit RNA sequencing or proteomics, estimate the transcription, translation rates, for example. Drug response modeling is another way you can actually use ODE for, where you can predict how inhibitors will affect the transcription of protein degradation. Synthetic biology help to actually design genetic circuits, predictable dynamic behaviors. It is very powerful because it actually captures continuous dynamics. It is mathematically well established. It's actually very helpful in small to medium systems. And it's really deterministic in its nature, very reproducible. On the other side, it really requires homogeneous assumptions. It really has to assume there are well-mixed systems, and it is very sensitive to the initial condition. So if you have a small error at the beginning, it will cause large deviation in your prediction eventually. It is not very scalable to large-scale genomic systems. So because it's computationally expansive and extensive, it's really hard to also do because of that part. Popular tools you can actually check. You have BioNet-Gen, it's a rule-based modeling. You have virtual cell spatial modeling, and Chase is another cell-based modeling. Cell Designer is one that is free for academic. It's very visual. Actually, you can model signaling, metabolic pathways. Does not require a lot of coding. So check these out and see which tool can actually help you in your research, in your hypothesis generation, and such. The third is physiologically based pharmacokinetics models, or PBPK. It's a special type of modeling that uses actual human anatomy or animal anatomy, physiology, biochemistry. It really simulates how drug moves through the body, from absorption to distribution to metabolism and finally to elimination. So the ADME aspect of how this, it's quite realistic because it actually compartmentalize the model into actual organs and tissues. So each organ basically or a tissue is a compartment connected by blood flow. And it has its own volume, flow rate, and such. It really mathematically relies on ODEs in a sense. You input a lot of physiological data, patient-specific data, drug and specific in vitro data, and the outcomes or the outputs you actually get is like the drug concentration, time profiles in plasma, in tissue. So it's very helpful in that way. It's quite realistic. It's not just curve fitting basically. It's just, you're actually simulating based on real data input. So you actually not predicting how it actually does. You're actually using existing data to fit on your model. It can simulate what-if scenarios, and it can actually integrate multiple data sources from preclinical to in vitro to clinical, and very helpful for personalized medicine. However, it's because of that, it's very data intensive. It really it's very complex, and any error can be propagated very quickly. So any mistakes in physiological assumptions or parameters will actually shift the outcome significantly. You can use it in actually PK in special population because we don't know how to dose in pregnancy, in pediatrics, in renal hepatic patients, in elderly. So it's helpful to actually simulate that. It actually can be used from scaling from animals to humans. So a mouse that is 30 grams is different from a human who's 70 kilograms. How can you actually use the data you have from an animal and fit it to a human with a different fat distribution, different muscle, different blood flow? It really is helpful to translate the data from animals to humans before necessarily doing a lot of human kind of testing. Drug-drug interactions is another way actually of using this. It's very helpful. Formulation endorsing optimization. How can I alter the molecule for better penetration, for better stability? You can use all these to actually test in these kind of in silico models. And for FDA and EMA acceptance, really actually do now accept these modelings as data is valid. Few public popular tools. PKSim is an open source used by pharmacies. SimKip is simulated that it's commercial, and so is GastroPlus. MATLAB SimBiology actually is another tool for you who code to actually use. And if you are an R person, R packages like MRGSolve or DSolve are helpful in custom modeling. But again, PKSim is probably the easiest one for us to explore a little bit if you want to stay open source. SimKip and GastroPlus are stuff that you can look at, but require a credit card and a big bill. So going back to the virtuous circle we spoke about a few slides ago, we are at the age where we have a lot of these omics that we have developed, transcriptomics, genomics, proteomics. We have extensive data on them nowadays, and we have computational pipelines to analyze these from gene expression to protein life cycles and degradation and such, that you can actually use for signature in outcomes and disease severity, and you can integrate from other sorts of data from these omics, create a new hypothesis, and do in silico research to confirm molecular genetics or do biochemical analyses, functional analyses. So you go from very global approaches to targeted approaches, understanding for a specific disease or a specific type of host, all that type. So we have multi-omic analysis really are able to combine all these omics data in one model, adding on them our understanding of a field that all of you are seeing growing exponentially in the last 10 years, the microbiome understanding, for us to understand biology more comprehensively than we did before, from just, a pathway of a Krebs cycle, but how does this disease happen? How does this disease alter a physiological function, and how that altering actually affects the disease severity, the response, the treatment. All these are stuff that we're now able to hypothesize and test in silico before we go and actually do any human or animal model studies. So it is very helpful for us to understand that you are adding pipelines on top of each other, and what you're getting is a completely different understanding of physiology, of biology, of pharmacokinetics, just from that presence of these multidimensional equations now coming all together in a way that we as humans cannot really see. I have this novel that I really liked. It's probably more than 100 years old now, called Flatland, A Romance of Multidimensions. And it talks about a two-dimension world with its objects presenting different beings, and then a sphere, which is a prophet coming from the third dimension to this world, and how they have trouble understanding who he is or what kind of world he's coming from. And this is the same for us. We are really unable to even imagine what the model is seeing from these 200 or 2000 dimensions, but we are able to confirm that these are real assumptions, valid data points that we can actually build insights on, or get leads to actually pursue additional research into pathways we never saw there, checkpoints that we never thought actually affect that process and such, just from these modeling in this in silico approach. And this is probably the most important part to understand about how this is now becoming different from it was before, in terms of how we approach and we look at biomedical research and translational research, where we are shifting to the network-based approach from the pathway analysis. So in the past, we used to focus on the pathway of any process that we actually are looking at, identifying the steps of it, the rate-limiting steps, the molecules that affect it positively, negatively, the byproducts, the outcomes. So we were very focused on a pathway-led analysis. Now, as we discover more and more of these, we are able to put together a network of analyses, or a network of processes together from different pathways, where this molecule is shared between pathway number 1, pathway number 2, and how that interaction affects one pathway versus the other, and what other parts of the network that are not necessarily part of the pathway might be playing role into shaping the pathway functionality. This is something that we would not have come to understand without these very strong, very complex mathematical models that we can now use network biology to understand things that might not come to us instinctively in terms of relationship between pathways and molecules, protein-protein interactions, protein-gene interactions. And I'll talk more about the interaction networks, because this is very important for us to understand. So interaction networks are the thing that is being unlocked by AI, and let's talk about how this is actually unlocking the new horizon for us for the future, and how this is approaching the limit with our knowledge as we have, in opening the road for machine learning and computational models to come and actually open a new horizon for us. So let's talk about a theoretical environment where we have 7 proteins and 5 different enzymes, and you are trying to figure out how many pathways you can actually create thinking about these. There are protein-protein interactions, and there are, of course, protein-enzyme interactions. So each pair can represent a pathway, a potential beginning of a pathway. So if you look at how many pairs you can do, you can reach 35 different pairing possibilities. However, sometimes you actually have more than one enzyme interacting on the same protein and its byproducts. Sometimes you have multiple proteins interacting with each other. So the pathway could include more than a pair of these. And if you are trying to map them, you can actually create a network with 66 components, each one of them representing a unique relationship between the two parts of this pathway. If you are trying to see how many longer pathways you can create, where we actually involve more than one step, in this small environment of 35 pairs of 12 components, you would actually be looking at more than 10 billion pathways. Imagine that. Now, of course, this is what they call a complete graph, which each potential pathway that is hypothetical is existing. The real world does not work that way. You know that there are proteins that don't interact with proteins, or there are steps that cannot happen. So whatever is not logical, if you take it out, you probably have somewhere in between the 66 and the more than 10 billion opportunities. And this is where the machine learning and computational power comes to help us. For us humans, when we look at the world, we see three dimensions. Sometimes they consider time as a fourth, but for the physical beings, we see three dimensions, X, Y, and Z. And we can tell the relationship to each dimension for an object by looking at it and saying it's closer to the ground, closer to the window, closer to the ceiling, through navigating the world with these three-dimensional perspective. Imagine a machine or a computer that can look at 200 dimensions and tell you this single data point has this relational distance to this dimension and this distance to that dimension, and be able to tell you what 2 or 3 or 5 dimensions it actually achieves the closest distance to in a hypothetical 200 dimension world. This is where the machine can actually come and be very helpful in forecasting and foreseeing stuff that we have not yet figured out from our three-dimensional view of the world. And this is where the power of like computational biology, computational physiology comes to the rescue. So unlike a pathway where you have a step to a step to a step to a step that is linear in its nature, you do have a network where one step from one pathway can actually interact and lead to a different dimension or a different pathway from a different set of pathways. So let's say you have a pathway A with 7 steps and pathway B with 12 steps, and pathway A and B actually interact intersect at step 4 or step 5 of pathway 1 and step 7 and pathway 2. You can walk from pathway A to B, creating a third pathway that you have not even thought of existence, or yet maybe had some evidence of it. You have some now computational relationship that tells you there is weight to this pathway that actually might signal something of a third pathway that you have not thought of. And this is what you can actually get from interaction network, where your nodes representing proteins, genes, metabolites, any complexes, and the edges is the potential relationship you can actually see when you look at the bigger population of these molecules together, and then actually discover potential relationships or pathways or whatever you want to call it that will guide you towards something you need to look for from the bigger components of this network. And you see here, when you actually present them with dots, which one is bigger, and with the lines, which one is thicker. That actually tells you where you need to focus your attention to, because there's something at least computationally that sounds like high probability or high yield. And to discern that, we have few types of pathways / network analyses that I'm going to talk briefly about. And I borrowed this very nice illustration from bioinformatics.ca. And we have 3 different modeling ways. and a few tools that they actually can help you look at them. The first one is the enrichment analysis of fixed gene sets. This is a very popular method used in bioinformatics to identify biological pathways or functional categories that probably are overrepresented in a list of genes that we have not kind of figured out yet. So it's typically derived from experimental data such as gene expression analysis or mutation data. And it's particularly useful for identifying the underlying biological processes or molecular functions associated with a condition or a disease. And it's a computational method used to determine whether a predefined set of genes shows statistically significant concordant differences between two biological states. Now, the second approach is the de novo subnetwork construction. And this is referring to identifying or building a subnetwork from scratch, a group of pathways together, meaning without relying on predefined lists or databases, just using data-driven approaches, the network analysis or network representation, you uncover condition-specific functional modules or molecular pathways. This is what I was referring to, the example of jumping from pathway A to B and creating a new pathway C, for example. It basically involves representing and analyzing biological processes as interconnected pathways using these tools and databases to understand the interaction between entities like proteins, metabolites, and also uncover insights into disease mechanisms that we have not really thought of, and potential therapeutic agents that also would have not been seen without this 200-dimension approach to this data set or with this environment. The third pathway is the pathway-based modeling, is another computational approach that used to understand and predict biological processes by modeling the interaction between genes, proteins, or metabolites involved in a specific biological pathway. It allows for the integration of multiple types of data in complex studies, like complex diseases, cellular behaviors, response mechanisms, and it's widely used in biology, very helpful in drug discovery, and hopefully going to be leading us to a more personalized medicine, the dream that we are been thinking about for 20, 30 years now, probably. Coming to the last part of this talk, which are some practical tools that I specifically find very helpful because they are zero coding, very visual, and I wanted to talk to them to see how we can move from vivo to silico research and then going back. Reactomes and KEGG. Reactome is an open source curated database of biological pathways, reaction, and processes in human. It's really been developed over years and years from the European Bioinformatics Institute. It is focused on molecular events, chemical reaction, signaling events, functional interactions. It's very, very diverse and very strong. And of course, it's all literature reference, so it's nothing that's being guessed that you're talking about. You have more than 2,751 human pathways, almost 16,000 reactions, more than 11,000 proteins, and more than 2,000 small molecules and 1,000 plus drugs, all represented that you can actually put together and see the interactions. And then, so it's an open source, peer-reviewed, highly curated database. KEGG is a collection of biological information compiled from published data material in databases, includes information from genes, proteins, metabolic pathways, molecular interactions, biochemical reactions, and such. So putting these together, you can actually have a significant insights that are new as we spoke. It provides a very nice relationship map. It can actually look at different components and the interactions. They both explicitly describe biological processes as a series of biochemical reactions, and they represent many events and states that are found in biology. And as someone who typically deals with research from the outer surface of the cell all the way into the organism and maybe the society, it's very interesting to actually play around and see these tools. And I'm going to show you next a slide of how Reactome looks. Very visual, very nice, and you can throw in all these metabolic pathways and add this function, alter this function, upregulate, downregulate, overrepresentation, and such. And it tells you what could potentially happen theoretically if you do that on the entire system that you are looking at. So very visual, very nice. This is from KEGG, and these are the different pathways that you can actually look at and put in this equation and see how, again, you alter the environment hypothetically and what actually happens to other components in these systems and other molecule states. I have a few other databases that you should probably check. Some of them I already mentioned, like BioGRID, IntAct, KEGG again, MENT, the Reactome we spoke about, pathway commons, wiki pathways. All these are very helpful for us to actually look at pathway in network database kind of integration. Very useful databases. If you are a coder, you can actually get the pipelines, put them together, and get something that is probably even more strong than Reactome and KEGG. De novo network construction and clustering. You have a few nice tools such as GeneMania, HotNet, Hypermodules, and Reactome Cystoscape app. I included all these clickable URLs for you to go and play around with these. I'll put it in the description for this lecture as well on YouTube. Again, for pathway modeling, we have a few platforms such as GeneMania, again, HotNet, Hypermodules, and again, Reactome Cystoscape app. All these are nice, visual, easy to use. A lot of them are for free, some of them are not, and as academia, I probably can ask and see if they have any special offers for you. As someone who does a lot of extracellular research all the way to the organism and beyond, seeing these intracellular mechanisms interacting, getting insights of the interaction between different pathways, different molecules is very insightful. Although I might not be using Reactome and KEGG on a regular basis, I really like to actually play around with them as part of my fun. Very insightful, very helpful for us to see how these microsystems affect the larger picture, affect the extracellular interactions and gene expressions and protein expressions and such. I encourage you all to take a look at them, even if you're clinical. It still could be like an opening for you to talk to your PhD colleagues on the other side, do a few research ideas, and get them to becoming really world-moving discoveries. I hope you all enjoyed this lecture. These are my emails to reach out to me with any questions, and I'm always happy to kind of learn more about this, get any additional insights, tools that I might have missed, or concepts like be these computational or equational that I should learn about. I'm always open to your feedback. Thank you so much and have a wonderful day.